and welcome to the... Sorry, Virginia, I'll wait till the recording has started, which it has now. So hi, everyone, and welcome to the Coastal Forage Fish Network's annual virtual symposium. We're really excited to have you join us tonight, and we're really excited to talk about all things forage fish. Just a couple of housekeeping items. All the participants will be muted on entry. So if you'd like to ask a question, please raise your hand or type it into the chat. We will hold all questions for each speaker to the end of each speaker. So if you do have any questions, please hold them till that time. Our wonderful facilitator, Virginia, will pull together all the questions that have been entered in the chat, and she will pose them directly to the individual speakers during the allotted question period. So if you're popping a question into the chat, don't worry about putting your hand up during the question period, because Virginia will take all of those questions and ask them directly to our speakers as time allows. If you'd like to ask a question directly, please just remember that you're muted. So feel free to raise your hand, unmute yourself when you're called upon, and then you can go ahead and ask your question. Just a reminder, your videos are also off automatically. So if during the question asking period, you'd like to have your face show up for your question to feel a little bit more personal, feel free to take your video, put your video on, just take the little cross off by clicking on the video icon at the on your screen, and we can go from there. If you have any questions, feel free to pop them in the chat. I won't be able to moderate the chat as I'm presenting, but like I said, our wonderful facilitator, Virginia, will be able to answer your questions for you. So thank you so much for joining us tonight. We're really excited to have you here. My name is Caitlin Pierzchalski, and I'm the Executive Director of Project Watershed, and I'm here today representing as the co-chair of the Forage Fish Network. I'll be hosting tonight's event alongside my co-chair, Kyle Armstrong from Peninsula Stream Society. So tonight on the agenda, we have an action-packed evening talking about our lovely forage fish and a couple of different parameters. So I'll provide an introduction to the Coastal Forage Fish Network, which is the group hosting this event tonight. And then you'll get to hear directly from some of the Coastal Forage Fish member groups. This includes PODS, Tsleil-Waututh Nation, and the North Island College. From there, we'll move into our focused researchers. So you'll be hearing from Jessica Qualley. She'll be talking about herring research in the Strait of Georgia, and Dr. Simone de Roches, who will be talking about the ecological variability as an indicator of shoreline restoration success. And they'll be talking about a decade of community science monitoring across the Sound. For the agenda tonight, I'll be speaking, I'll do an intro for 15 to 20 minutes, and then each of our member groups have 10 minutes for their part, which includes a section for questions. So if you'd like to ask questions for any of those individual members, please remember you can use the chat or hold your questions to the end and pop up your hand, and we'll call upon you. Depending on how time goes, we may have an optional break and trivia at about the halfway point at about 5 to 7. And then right at 7, we'll be jumping into presentations from Jessica Qualley and, of course, Dr. Simone de Roches. Kyle will be facilitating that second half. So if you have any questions, Kyle will be presenting or launching into the presentations for us at 7 p.m. So very excited to have you here. So first and foremost, what's the Coastal Forage Fish Network? The Coastal Forage Fish Network is kind of the current iteration of a bunch of different member groups who have been participating in research on forage fish through time. And then most of this research has a very community science base. First and foremost, I'd like to acknowledge that this research has been happening for a really, really long time. And we're here today building off of the legacy of past works. So the Coastal Forage Fish Network is a relatively newly established group that's been around for 2 years under either that name or a slightly different one. But the work of this network has really been built and launched through time. Specifically, I'd like to really acknowledge the efforts of members and founders through time, including Ramona DeGraff, Jacqueline Bars, and others from WWF. This work was really built on a legacy that came out of research that was happening down in the States, and then some really keen researchers, people from the community, and interested scientists went down, learned from their work, and basically brought those methods up here to Canada. And those are still the basis of the work we're doing today in a lot of cases. So as a network, we're really building off of the work of many people before us. And I'm really excited to be here today representing as the co-chair of the Coastal Forage Fish Network. But I really do just want to acknowledge that this has been a process that's been built over the last 15, almost 20 years. And we've been working really, really hard to further that work. And now we're here together as a little bit more of a facilitated group, being able to share the work of many people through time and many member groups through time. So as a group, we're stewardship groups, nonprofit organizations, coastal First Nations, and all sorts of folks, community members who are dedicated to the study and conservation of forage fish in BC's coastal waters. As you folks very much know, forage fish can be a lot of different species, but they represent a really core piece of the coastal food web, something that supports many different charismatic megafauna that we're all very fond of. Most of our focus through time has been on some of the shore spawning forage fish like surf smelt and Pacific sand lance, because that's been one of the methods that we've been focused on for detection. So we do a lot of intertidal beach surveys that do visual detection of eggs. And so a lot of our work has focused on those two species that are shore spawners, spawning in that intertidal area. As a group, our goal is to share information, knowledge, and resources. We undertake collective research and share that information out. And we also help support new groups in getting forage fish monitoring up and running in their regions. So there's many groups participating, sharing to a large database of information so that we can understand some of the gaps that exist about these species, and then share more knowledge about them as well through time. So we've had a huge time of growth and establishment, which is really, really cool. We have a huge list of participating members, of which you can see the list here, but I'm actually going to flip us through to a map, which will show us a little bit more, better represent and show you visually who we are and who's participating in this research. Currently, the research covers coastal British Columbia through the Sunshine Coast, Vancouver Island, Howe Sound, Burrard Inlet, the west coast of Vancouver Island, and the lower mainland. Eventually, we'd really like to expand our support for forage fish monitoring, and we'd love to have the full breadth of coastal British Columbia represented by our efforts. Just a big shout out to so many current members. We have the Friends of Semiahmoo Bay, Loon Foundation, Mount Arrowsmith Biosphere Region Research Institute, Parks Canada, Peninsula Streams, Project Watershed, Redfish Restoration, Sunshine Coast Friends of Forage Fish, Tsleil-Waututh First Nation. We have so many different participating members throughout the region: North Island College, Conservancy Hornby Island, Qualicum Beachkeepers, and every different group represents a little parcel of the area. And as such, by doing this in a really kind of like regionally distinct way, we're able to represent collectively a really, really broad breadth of region throughout coastal British Columbia, which is really exciting. So we've had a huge year for growth, which is super exciting. We have done a lot of things in the past year that we would really like to celebrate. Specifically, we had a name change, which is really exciting. We started as the BC Forage Fish Monitoring Network, which was a little bit of a mouthful. So we reduced our name to the Coastal Forage Fish Network. So if you've seen a little bit of change from the BCFFN, now we're the CFFN. So just a little bit easier to handle acronym-wise and title-wise, so that we're all still the same thing. We've received a big funding announcement because of BC Shrift, so we've got a lot of funding now to support the network, which is really exciting. It's been able to support us in growing our capacity as a network, as well as supporting new groups, equipping groups, and doing all sorts of stuff to help support the network. In partnership with Peninsula Stream Society and DFO at the iOS facility, we hosted an all-network training event at the Institute for Ocean Science in Sydney. And we were able to do a big review of our field sampling and how we collect and make sure we're all on the same page and really optimizing our collection. And we also went and did a great egg identification workshop with our DFO colleagues who are doing some great work as well. So it was great to learn from them and share information with our colleagues. We've also expanded to the west coast of Vancouver Island, and we are focusing on an eDNA collection program there to expand the habitat suitability model, which was developed for the Salish Sea. We've also launched an iNaturalist herring project called the Pacific Herring Spawn and Nurseries Project. If you would like more information about that, you can definitely go and check out iNaturalist, and we'd be happy to share the link of that project there. We went from having very few observations to having like so many more this year, so that's really exciting. The aim of that is really to visually track eggs and milt in any areas where our wonderful community scientists are observing them and then uploading them to iNaturalist so we can track those observations through time and space. Furthermore, we had our inaugural Pacific Sandlands Day, which is still absolutely unofficial, but very exciting. Official to us, unofficial on your calendars. But that was hosted this year, December 8th. And the goal of that is really to coordinate across our groups so that we can have kind of these discrete moments in time where we're all going out and sampling in tightly clustered time, like temporally, so that we can really see like, when are we seeing these things? Where are we seeing these events? And those tight clusters of time really help us to showcase when we're starting to see spawns specifically for Pacific Sandlands and what that looks like across the region. So these tight temporal clusters really show us cool things. And that was kind of the goal of Pacific Sandlands Day, and also to give us a little bit of celebration and to celebrate our tiny little fish. We have hosted 6 member meetings to coordinate our efforts and share resources. We've equipped and trained a bunch of groups, which is really exciting, and that has been made possible by our BC Shrift funding. We have engaged 5 new member groups this year and have 5 affiliate members as well. We also have established through PSS an agreement with DFO to do egg validation, so to support our identification process for our visual surveys. I just wanted to show a couple great photos of the folks in the field who are really doing this work. I get to stand here and talk about it, but these are the people that are really, really making it happen, which is super exciting. So these are the lovely folks from pods out in the field. You can see some sieving and vortexing happening here, which is awesome. This is a key part of those visual identification surveys. Here we are again on the beach at Lambs Bay with the Friends of Forage Fish from the Sunshine Coast. They're doing that sieving and vortexing process, and you can see they're all out here on different beaches. These are the support of community scientists and volunteers. And here we have some training with science volunteers at the Friends of San Diego Bay and Mabry. So these are the lovely folks training up new volunteers and showing them our methods. So this is a great example of how we engage folks and how we show them how to do these visual ID surveys and make sure that we're all using the same methods so we have comparable data, which is awesome. And here is an example of our egg identification workshop that we hosted at iOS. So here was us going through some of the sampling and sieving and vortex methods just to make sure that we're all using the same strategies and that we're kind of optimizing as we can. People who have been doing this a lot have different different little optimization tricks. So it was an opportunity for us to share those with each other. And here's Moira teaching us how to ID all the forage fish that we might be seeing in our samples. It was great because we looked at different eggs and how they look through their growth and development. And then she also showed us examples that you can see here of the larval stages of the fish as well. So here is like a summary of effort, which I think is so cool. Over the past year, from April 2023 to March 2024, we've accomplished a lot. We have 24 organizations, 5 of which are newly trained, with 392 volunteers. They put in 3,489 hours doing these detection surveys. We collected 686 visual sand samples and 253 eDNA samples across our entire network. The Pacific sandlance eggs or eDNA were found in 98 of these samples, which is a total of 14%. And surf smelt eggs or surf smelt DNA were found in 56 of these samples, or 8%. Oops, sorry. I scrolled through my notes and it took us too far. And then of these, this year we launched our first herring observation program using iNaturalist. So far we have collected 281 observations of Pacific herring spawn using that survey. Finally, it was also the first year we rolled out Pacific Sandlance Day. It occurred on December 8th at the height of the sandlance spawn. So those are some of our kind of key highlights. We're really excited to showcase the efforts of specific members. So I'll be flipping it over from this point. I'm going to stop sharing my screen and then allow some other folks to share. So at this point, I'd like to flip it over to Sydney from Pods to talk about their work.
Awesome. Thank you, Caitlin. All right. Share my screen here. Oh, of course, it has disappeared. 1 second.
We could kill time with a trivia question. Virginia.
Just to break the ice, does anyone know what a fish's favorite radio station is? The Current on CBC.
Thank you, Virginia. That's awesome. I'm also just realizing that I actually forgot to recognize and do our territory acknowledgement. So in order to kind of start us off in a good way, I would like to cycle back to that. Thanks everyone for your patience, and just recognize that from here, where I am in the Comox Valley, we work for the most part in a very regional way, and we work on the traditional territory of the traditional and unceded territory of the Comox First Nation, which of course represents a group of collective of other nations as well. But also recognizing that the group of the Forage Fish Network happens throughout coastal British Columbia, which is the territory, the traditional and unceded territory of many different nations. So as participants, it would be great if you could kind of jump into the chat, reflect on the territories from which you are coming to us. And if you'd like to, you can pop in the chat on those traditional territories and just recognize the areas that we are so lucky to work, live, and play. And at that, over to you, Sydney. Thanks so much.
Awesome. Thank you, guys. So good evening, everyone. Thank you all for being here. My name is Sydney Flummerfelt, and I work and live in the Seychelles Swia. I work for Pods under Jen Blankard, who is the field research supervisor for the Loom Foundation. Today, I want to be giving you a review of the work we've been doing within the Seychelles-Souillac and our recent expansion into Sliaman territory. So for those of you who don't know us, I'll just give a quick background of the foundation. So PODS, the Pender Harbor Ocean Discovery Station, is part of the Loon Foundation. The Loon Foundation was established in 2002 as the Ruby Lake Lagoon Nature Reserve Society, and in 2020 changed to the Loon Foundation. In 2017, we started monitoring forage fish spawning as part of our Pender Harbor Coastal Waters Monitoring Program. So here's where we are located within the Salish Sea. This is Pender Harbor on the Sunshine Coast, and this is where most of our forage fish monitoring has taken place since 2017. And this is where we were able to expand to in 2023. So this is Powell River and Texada Island, which are located within Sliaman territory. So in Pender Harbor, we started off with 4 main core sites. You can see them there in yellow on the map. And this also includes our sites on Thormanby Island to the south of the map, not just our Pender Harbor sites. So in the winter, when we have a confirmed spawning at one of our core sites, we start blitzing other sites in hopes of identifying new spawning beaches. So we have surveyed over 27 blitz sites and over time have identified 7 spawning beaches within the region. So we were really excited this past winter to be able to expand our program to include sites within Sliaman territory. Thanks to the Pacific Salmon Foundation, we spent 4 days surveying Powell River and Texada Island, and we were able to complete 14 surveys at brand new beaches. In Powell River, we went to 12 sites. However, one of them, Kalani Beach, we decided didn't look like suitable habitat, so we didn't survey there. And Emmons Beach, we went there knowing that the tide would be too high to complete a survey. However, we did want to go there and scope it out just for future surveying. And then Penrose Bay, we unfortunately could not gain permission to access it, as we would have had to cross through private property, and we just weren't able to get the permission that we needed to, so had to move on from there. But from the 12 sites, we were able to sample 9 of them, and of the 9, 5 of them were positively identified as Pacific Sandland spawning sites. So we were pretty excited about that. On Texada Island, we viewed 6 sites. Although we did attempt to get to 7, Texada was a bit tricky since we only had a 4-hour window to sample our sites, just due to ferries and our tide window. So we decided we'd start at our farthest sites, which were Pilings Beach and Cook Creek Beach. Texada is very big and most of it is dirt logging roads. So it was quite an adventure for us in Jen's little RAV. But we made it to Pilings Beach, and when we got there, we realized that the beach was down quite a big hill and there was no trail to get there. But from what we could tell from the top, it didn't look like the most suitable spawning habitat, so we decided to keep on moving. We continued down the dirt roads, and when we thought we were getting close, we unfortunately ran into a locked gate, and sadly, that was that for us, and we couldn't continue, and we had to turn around. Luckily, we were able to sample all of our other sites on Texada Island. We sampled 5 sites, and 1 turned out to be a spawning beach, so that definitely cheered us up. We had a lot of fun doing all the sampling. We are really excited about how successful we were in identifying new spawning beaches within Sliaman territory, and we look forward to continue monitoring. We really want to continue monitoring the positive beaches we've identified We want to survey new beaches, including the Emmons Beach that we already scoped out. And then we're also looking forward to getting permission, hopefully, to survey Cook Creek and Penrose Bay, especially the Cook Creek one. And we're also hoping to recruit and train local groups and volunteers. So if any of you are from the region or if you know anyone, definitely let us know. We're always happy to get more involvement. We were able to have one volunteer from the local naturalist club come and do 2 of the Pell River sites, which is awesome. We're hopeful to recruit and train more volunteers next year, as well as connect with the Slammin members and guardians and get them involved in our surveys. Unfortunately, we were in contact this year, but unfortunately we weren't able to have them come out due to illness, as well as the tide windows made our surveys at inopportune times. So it just didn't work out this year, but it's definitely something we're looking forward to next year. Yeah, and if anyone is interested, all of our data can be found on the Strait of Georgia Data Center. So definitely check that out if you're interested. Awesome. So thank you so much. And thank you to everyone involved in making this happen, because it's always very exciting to share what we've been up to. So thank you. If there are any questions, please ask away.
Great. Thank you, Sydney. I'll start us off with a question, which is, how can we reach out to you if we want to volunteer in your area?
Yeah, that is a great question. So you can reach out to Jen Blankard. Her email is on our website, and also I think everyone within the CFFN has it as well. So yeah, you can definitely reach out to her, and she can let you know when we'll be there and when our we won't be there until I think December. But yeah, definitely reach out anytime and we could get there.
Awesome. Thanks so much, Sydney, and thanks so much for sharing your work.
Yeah.
I'll just look over to the chat now and see if there are any questions. So feel free to pop them in there. Virginia, if there's any hands, feel free to point them out for me. And we can go from there. I'll just say that personally, 5 new beaches in a year is super exciting. I'm just really stoked for you folks and stoked at the effort that you go to get all these new beaches. So a big congratulations for just being so ambitious with your approach and being able to identify those 5 new spawning beaches. That's super exciting. Right on. Well, if that's it for questions, I'm not seeing anything. Jen was great enough to pop her email in the chat, so it's there for anyone that would like it. And if that's it, maybe Sydney, I'll get you to pop off your screen share, and then I will pass it on to Graham from Tsleil-Waututh First Nation.
Awesome. Thank you so much.
Wonderful. Well, thanks everybody. I'll just get set up here. Put in the presenter mode. Okay, just okay. Well, this is kind of I'm excited to be here and follow up with that because yeah, I'm actually living up in Tla'amin territory, and Mahud's Beach is just right down the street from me. So maybe we should get in touch and can help volunteer in the coming years. But yes, hello, I am Graham Nicholas. I work for Tsleil-Waututh Nation as a senior environmental specialist. And this is more, I may have misread the email, but this is less of a yearly update for our field programs and more of just I guess, a presentation on the history of forage fish in Tsleil-Waututh territory. Yeah, I am a non-Indigenous biologist or ecologist, whatever you want to call it. I've had the privilege to work for the nation now for about 5 years. And so what I'm going to share here is certainly perspectives from Tsleil-Waututh. They might not represent other nations, but sort of just the teachings and things that I've learned along the way and I'm okay to share. So to start off, let's just situate us where Tsleil-Waututh territory is. It's, you know, Burrard Inlet is where we're talking. So Vancouver, incredibly urbanized area, a lot of history of colonial development. But really, I wanted to highlight this map to show that there's a long, obviously, history of habitation in this area, and you know, many seasonal village sites existed throughout these waters, as well as sort of long-term village sites. And a lot of the seasonality of villages and moving was certainly based on resources and what was available during the time or time of year. And so Tsleil-Waututh are very much people of the inlet. You know, the word Tsleil-Waututh is what the, you know, collective word for Burrard Inlet is. And so Tsleil-Waututh, you know, historically pre-colonial times had a, you know, 90% of their diet came from the ocean, with sort of four main staples, salmon being a major one, forage fish, including herring and other species, clams, and marine birds. And we've certainly gotten away from that in terms of diet, but I think it really speaks to just, you know, how intrinsically connected the nation was to its marine resources. And so present day reserve is here, our number 3, and so certainly squeezed into a very small area from what their historic sort of area would have been. I think it's really also impressive to note, you know, everyone thinks of salmon as being such a, you know, a staple of Indigenous people, and through some of the research that the nation's been undertaking in terms of looking at, you know, pre-contact food webs through the archaeological record, it's noted that, you know, forage fish rivaled just as much as salmon for a diet. So it really speaks to the importance of forage fish to Indigenous populations. And this probably would have been throughout the coast, and every nation probably would have, you know, been collecting forage fish for consumption as well. And so I'm just going to sort of go through a little bit of a history here. And this is research that's been done through our Cumulative Effects Monitoring Initiative, which is really trying to understand what pre-contact conditions were and pre-contact food diets were. And I like this is an interesting map because I like to think of it as, you know, we think of ecosystems like a healthy ecosystem that's complete. You know, all species are, you know, contributing to something or other. And a big thing that I think in ecosystem restoration is we tend to not include the human aspect of ecosystems, and Indigenous folks would have been just as part of an ecosystem as sort of a salmon were to orcas, for example. And so I think that's something really important to keep in mind, that, you know, humans are also part of that ecosystem. And if we think about like how long Indigenous cultures persisted and how sustainable they were for thousands of years, it really tells to the stewardship of Indigenous folks and the relationship to species and food. So as we get through sort of this timeline, we see like the early contact of colonial folks into the region, and this is specific to Burrard Inlet, this example, and it's certainly it's not a very, it's a illustrative example here, so don't take this, well, it's accurate to as much as it could be. Sorry, I've got a cat jumping up here, wants to join on this presentation. So there's been some major shifts that happened really quick, and one of the big, you know, things was the collapse of herring, and it's a really interesting story. I mean, it's a devastating story, but it began in the late 1800s when there was an intense fishery for herring, where they had a floating refinery called the Spratts Oilery, which basically rendered herring for its oil, to use that oil to grease the logging equipment, the skid roads, and all of the equipment that was responsible for logging the North Shore Mountains and the surrounding area. So it's it's interesting that they, harvest one species to then affect and impact, a whole ecosystem. And the herring were so abundant that they actually used a dynamite fishery to harvest. And it's exactly as it sounds, like you throw dynamite in the water and you get a lot of herring at once. And so they would, scoop up these herrings, render them for the oil, and they would also then throw out all the byproduct into Cole Harbor and into the water. And so it was not just, destroying these populations at mass, but it's also that byproduct which probably, polluted the waters and, and discouraged other fish from coming back. And this was an incredibly, effective fishing technique, because they extirpated herring from within Burrard Inlet within 5 years. And so this mark is basically by 1885, there was no more herring in eastern Burrard Inlet. And then further to industrial exploitation, there was a massive oil refinery spill in like the late or early 1912, where it basically destroyed clam beds. And so as you lose herring and other species, they go after the smelt. And then those were further then extirpated and overfished. And it's just kind of this continual, go from one species to the next, just over harvest and not really think about the future. And then, you've got the shellfish closure, which we're not here to talk about shellfish, but it's just another example. And so you get to where we are presently, and, it's this, dismal populations, there's no more catch. And so we're in this state where things are not so great. And so a lot of the information gathered through this cumulative effects initiative, you're gathering information from archaeological record, pretty poor past records from commercial fisheries. And so you have to rely on everything you have, including traditional use studies, which the nation's been a bit a part of. So you're really kind of painting a picture, every little bit of the puzzle to figure out what it is. And we're really is what trying to say is, like, we're at the baseline that we have right now is not even comparable to what it would have been. And so we might think that things are doing okay, but really, how okay is it? And I mean, that's the whole topic of the shifting baseline syndrome. And one, local example that I always think about is with pink salmon in the Indian River, where some years we're seeing returns of a couple million fish, and it's really exciting. It's amazing to see that, but then it just makes you wonder, well, this is probably just a small portion of what things used to be. But it's not all doom and gloom. You can see there is a small uptick here, and we are currently documenting and witnessing sort of the return of some of these species, which is incredibly exciting to be a part of and witness. And so it wasn't just the overexploitation of species, but it's also the loss of habitat that these fish rely on. And so here's an aerial photo of Vancouver looking east, Stanley Park, and so this is before seawalls were put in. So you probably have natural intertidals, less erosion because of seawall effects. You've got no Lionsgate Bridge here. You see West Van. I'm sure most people don't even know that West Vancouver is giant mud flat. So a lot of habitat loss there, and just the industrial ports that just kept building and building. And as you get closer to the east towards where present day Tsleil-Waututh Reserve is, here's what the second Narrows looked like in the late 1920s. And then in the distance here's the Maplewood mudflats, if folks are familiar with that. And present day Tsleil-Waututh Reserve is just right here. And so it's kind of this that 1-2 punch of you're just over exploiting at an unsustainable rate of species, and then you're also removing all their habitat. So you really get squeezed out, and it's a really unfortunate situation. And it's it's sad because indigenous cultures are built on species, and a lot of those cultural practices are related to certain species. And when you lose those species, you also lose some of that culture and the cultural practices, and not to mention the history of colonialism and residential school and all that. And so it's just it's hard to hang on to some of that at some points. And it's just, you know, if the species doesn't return, then that cultural practice may never return, you know. And so it's really important to work towards ensuring that species are there for, you know, future generations to actually, you know, learn and participate in their culture. So super important to the nation to document. And so that's kind of just a really brief where we're at now. And so you fast forward over 140 years and you know, more modern day stewardship, and for forage fish, it really kicked off, I think, with some past research, first identifying what suitable habitat is left, and early works, I think, done with Ramona and through WWF to identify where are the suitable habitats. And so through that, we then linked up with this network here and got training and started doing a lot of the visual surveys. And we had been doing that for about 3 years, and until capacity issues with our field crew, we just, you know, had to prioritize, you know, what's more, like, this is important work, but other stuff is sort of, we always have to every year prioritize what we're going to do. And so we decided to take a pause from the actual visual detections, but we're still doing a lot of eDNA sampling specific to forage fish, just through other projects. So we're just sort of in a pause moment from the visual, but we're still actively collecting, and with our field crews who are basically on the water year round. It's really, you know, both putting the face of the nation on back on the land, but, you know, we're also, you know, being present and being there. You witness a lot. And so through some of our other programs, such as our beach seining program, where we're targeting the outmigration of salmon, we generally pull up other species such as sand lance and herring. And this is really exciting because this is what we think, and we've gotten some DFO experts that are pretty sure that this is a little ulican larva that was caught out of Port Moody. So that was really exciting to see that. And so it's these little things where you just get really, you know, encouraged, and like, wow, species are still here, but we're still trying to pinpoint exactly, you know, what are the most important beaches and habitats for protection or restoration. And yeah, as we are out and about, you know, we witness a lot. So here's video of some Pacific anchovy that we occasionally see. They're shiny little gill plates, and some of the spawn in one of the islands up the arm. So it's encouraging to see that. And then also incredibly exciting is the return of herring. So over 140 years of, you know, a species extirpated, they're finally returning to spawn. And so that's, you know, I can't stress enough how exciting that is for Tsleil-Waututh to actually see the return. And we're so actively monitoring and really focusing a lot on understanding, you know, where they're spawning and the numbers and what goes along with that. So yeah, I mean, it's a good story to you know, there is positives. Ecosystems are resilient and fish are very resilient. So it's just a matter of ensuring that they have a place and they're uninterrupted to do their thing. So there's some active spawners a couple of years ago just depositing eggs. So again, just really, really exciting to witness. And it's sort of about all I'm going to say with this, and just wanted to thank everyone for their time, for listening, and always the support for the network for supporting all of our work. So thank you so much.
Awesome. Thank you so much, Graham. That was super interesting. And thanks for the great visuals. It's always just so awesome to see these fish out in their habitat, doing their thing, especially in such an urbanized area. Awesome. All right. Once again, quick request for questions in the chat or hands up in the participant list. So please let us know if you have any questions. And big thank you to Graham for coming out tonight and joining us, and for sharing your folks' work. All right, Teresa, I see you have a question. Please go ahead. You can unmute yourself.
Hi. Yeah, I was wondering when Graham first noticed the herring coming back to Burrard Inlet, and have they been watching for volume, etc., since it did first come back?
Yeah, so the first major sort of spawn where you can actually tell like there's herring in the water was in 2019. And since then, every year we organize dive surveys to actually, you know, see exactly where they're spawning and get estimates of female spawners and eggs. So every year we're growing and learning more. And it's not to say, I guess, that they weren't spawning before. It's just when we first noticed, a real, noticeable spawn. And every year seems a little bit different, timing wise. And we're really trying to understand, are these, is this population a bit different from sort of the False Creek or up in Howe Sound, because the timing seems to be a little bit different. So we're learning. It's all very fresh, but generally 2019 was the big year.
Have you noticed if the spawn's getting larger, or do you have a quantitative analysis of that since 2019?
It's hard to say. It seems to be just initially. Every year seems a little bit bigger, but we haven't fully looked at all the data comparable. And I mean, it's such a short time frame realistically, but it's encouraging. We are seeing, more activity and orcas returning in sort of every other species. So it's exciting.
Wonderful
so
news.
It continues.
Yeah, wonderful news. Thank you.
You're welcome.
Awesome. Thanks so much. Just in the caution of time, I'm going to flip it to our next speaker. And if you have any questions, hopefully we'll have a little bit more time at the end. And so maybe we can pull Graham back in if he's around still, and maybe ask some of those questions. And if not, we can follow up via email. So definitely let us know what your questions are so that we can pass them on. Thanks you so much, Graham. And now at this point, I will flip it over to Aaron at North Island College to share with us his work.
Beautiful. Everything looking good?
It is on my end. Please go
Perfect.
ahead.
So hello, Coastal Forage Fish Network biologists, citizen scientists, and community members. My name is Aaron Schmidt. I am a second-year biology student at NIC, North Island College. And this evening, I'm going to be giving you guys a presentation on the effectiveness of using chemical dyes to detect Pacific sand lance eggs in beach sediment samples. This experiment was done with the Biology 250 Directed Studies course with Supervisor Georgie Harrison overseeing this experiment. So before I start, I would just like to give a quick land acknowledgement and say that I am honored to be living, working, and learning on the unceded traditional territory of the Comox First Nations. This project was also possible due to the partnership between North Island College's Center for Applied Research, Technology, and Innovation and the Comox Valley Project Watershed Society. So Pacific sand lance are a key species in the marine ecosystem. They are an important member of the food web, not just for other fish, but for marine animals like birds and whales. They spawn intertidally, and identifying their spawning beaches is the first step towards learning on how to better protect and steward these important species. But there is a bit of a challenge in detecting their eggs in beach sediments. So the traditional method of examining non-dyed eggs is to collect beach sediment from the high tide line, then sieving and vortexing it to the appropriate uniform size, and then looking at these subsequent samples under a dissecting microscope for the Pacific sand lance eggs. This is a difficult task as many eggs are nearly transparent and they do blend in well with the sand. So as you can see, there's very clear eggs, and sometimes the sand sediment is very clear, and it does lead to some confusion and difficulty with this detection. So I myself volunteer doing detection with these eggs with Project Watershed. And while doing this, I got to thinking and asking myself, are all these eggs being visually detected using the dissecting microscope? And would dyeing these eggs improve accuracy of detection? So from these questions, I did end up forming 3 hypotheses. My 3 null hypotheses were egg detection will remain the same across all treatments, that there will be no difference in egg detection from 2 similarly trained and experienced observers, and that there will be no difference in egg detection rates between the observers and the dyes used. This experiment was conducted in the North Island College's biology lab using standard lab equipment, although a lot of this equipment can be substituted with items purchased at your local supermarket. The sand used was collected from Seal Bay and was sieved to the appropriate size. For the dyes, a total of 6 milliliters of red food coloring with the active ingredient erythrocene and 6 milliliters of 0.3% methylene blue was used as well. The Pacific Sandlines eggs were preserved eggs that was donated by Project Watershed. So 3 treatments were replicated 3 times per observer, with 2 observers used in this experiment. For the control, about 20 milliliters of sand, 200 milliliters of water, and exactly 10 eggs were added, swirled to ensure that there was an even distribution of eggs in the sediment, and then it was rinsed in a coffee filter to maintain uniformity amongst all the 3 treatments. For the food coloring, the same amount of sand, water, and eggs were mixed as well, but before swirling, 1 milliliter of food coloring was added, and then the solution was swirled and allowed to set for 24 hours before rinsing the sediment in a coffee filter. The sediment was rinsed until the water ran clear out of the bottom of the filter. For the methylene blue, the same steps were applied as with the red food coloring, but the dye was only allowed to set for 15 minutes before the rinsing began. So with the detection, a Petri dish was marked with a bullseye grid pattern with each parallel circle approximately 25 millimeters apart, and 2.5 milliliters of sand from the sample was put onto the Petri dish and observed under the dissecting microscope. With a maximum of 2 minutes allotted per dish, the observer would attempt to find the eggs with the dissecting microscope. If an egg was found, the timer was paused, the egg was placed onto a Bogarov tray, and the timer was resumed when the observers started looking at the dish again. This was done until the allotted 2 minutes was up. The sand in the dish was then disposed of, and the search would continue with the next 2.5 milliliters from the same sample, with a total of 8 dishes that were observed per sample. When the sample was completed, the observer would record the total number of detected eggs found, compare it to the 10 that were put in there, and move on to the next sample. Now you can see with this bar chart, it shows the mean detection ratios and the standard deviation for each treatment. And we can see a clear difference between the mean detection ratios between the control and blue dye, as well as between the control and the red dye. But there was no significant noticeable difference between the red food coloring and methylene blue. The test that was used to see if there was a statistically significant difference between the means of the eggs detected in the different types of dyes and with the different observers was a 2-way ANOVA test. As you can see from the values obtained, the results here, that the p-value was less than 0.5 was found for amongst the dyes. This indicates that the mean egg detection rates were statistically significantly different among dyes, and that there was a strong evidence that these results were real and not just a random variation. There was no observed statistically significant difference found between the observers and eggs detected, as you can see with their higher p-values, as well as there was no relationship found between the dyes and egg detection rates for each observers. A box plot was generated from this data, and it clearly indicates the differences in egg detection ratios amongst the treatments. The control is statistically significantly different than both the blue and red dyes, but there is no statistically significant difference between the blues and red dyes. So I did put all 3 eggs, the control, the methylene blue, and the red food coloring into this dish, 10 of each. You guys are more than welcome to see if you can spot them. I myself can see a couple of the blues and the reds. So in summary, the eggs did successfully take up the dyes, and the eggs dyed with the red food coloring appeared easier to see against the sand than the methylene blue, but there was no significant difference between the two. Statistical difference, eggs were also observed to be easily seen with the naked eye when dyed, as you can see in this lovely photo, and this could expedite the simple detection of presence or absence of eggs. This opens up further researching for testing other dyes, such as fluorescent dyes, and using UV lights to detect the eggs, and also testing the feasibility of doing this detection method in situ, so in the field. So thank you all for listening to my presentation. I would also like to give huge thanks and acknowledgements to my fellow North Island College students, Sam Schneider, Melissa Roberts, Sam Clark, Amber Erbshaws, and Taylor McManus for helping me throughout the year with forage fish sampling. I would like to also thank North Island College's faculty and staff, Georgie Harrison, the supervisor and instructor in this project, for all her help and support, as well as Naomi Tabata, the manager of the Center for Applied Research, Technology, and Innovation, for funding this project and making it possible. I would also like to acknowledge and give a big thanks to Project Watershed and its members, Virginia East, Jack Herard, and Caitlin Pierzchalski, for their continuing support and contributions to this experiment. Thank you, guys.
Thanks, Aaron. Yeah, so cool to see your work and to see it all come together. I really love that last photo with the Petri dish where you can very easily visually see the difference. So that's great. We do have a couple minutes. I think I could probably field 2 questions. So please feel free to hand up and unmute or pop a question in the chat if you have one. Jack Huard, please go ahead.
Hey, awesome work, Aaron. That is so great to see those little red eggs popping out, just as we hoped. What is cheaper, the red dye or the blue, the methyl blue?
So the red food dye is cheaper and it can be found at pretty much any supermarket. The methylene blue, which is kind of funny, you can find it at most pet stores that sells aquarium supplies.
Oh, okay.
But the red food coloring is definitely cheaper.
Yeah, do you think some of the groups who are watching right now could take up this method next time they're doing this if they wanted?
Oh yeah, by all means. That's something I'm hoping I can start testing next season, is start doing the in situ, like do this out in the field.
Right. Okay.
Awesome. 2 more questions here. There's a question from Jacqueline Barr about if there is a toxicity element to think about when using the dyes.
So
something you've thought about? Yeah,
that is something I did think about. We were going to use malachite green. It is another dye that was used in the past, but there has been there has been some literature that does show it is toxic and it is really hard to remove from the environment. Luckily, the red food coloring is not as toxic. We use it in our food and every day, and the concentrations aren't that high. And it's the same thing with the methylene blue. A lot of people actually use it as a disinfectant and a way to sterilize their aquariums at home.
Great, thank you. And one more question. I said 2, but I think this is a quick one. And it's from Anne Parkinson. And it's, what preservative did you use before dyeing?
So the eggs that we had from Project Watershed, they were all preserved in ethanol. Rinse the eggs thoroughly with distilled water, though, before we used them in this experiment, because we wanted them to be as natural as we'd be finding them out in the wild.
Awesome. Well, thanks so much, Aaron, and congratulations on all your hard work. It's so cool to see the result together like this. So thank you so much for your ongoing commitment to forage fish and for pulling together this great work.
Thank you, guys.
Awesome. So as sort of predicted, we don't have time for a break probably. So I am just going to flip it over now to Kyle Armstrong, who's going to introduce our 2 next speakers.
Right on. Yeah, I have the pleasure of introducing 2 really amazing scientists, and starting with Jessica Qualley. Jess is a project manager with the Pacific Salmon Foundation and a master's student with the Francis Juanes at the University of Victoria in the Juanes Lab. Jess is interested in the factors controlling the migration ecology and summer distribution of adult herring and the importance of Strait of Georgia herring to the marine food web. Welcome, Jessica. Take it away.
Hey, thanks. Oh yeah, I'm unmuted. Thanks, Kyle. Let me just share my screen. Okay, can everyone see that all right?
Yep, we sure can.
Great.
Okay. Yeah, so great to see all of the great work that you guys are doing. I'm Jess Qualley. I'm a master's student with Francis Juanes at the University of Victoria. I study adult herring migration patterns. I'm also a project manager with the Pacific Salmon Foundation on a new herring project that's been funded through the BC Salmon Restoration and Innovation Fund. Jack asked me if I would speak today. It sounds like the Coastal Forage Fish Network is tiptoeing into the herring world. So I thought featuring this new BC Thrift Herring Project was a good way to talk about what we know about herring, what we don't know about herring, and how this project's trying to address some of those knowledge gaps. I'd first like to acknowledge and respect the Lekwungen peoples on whose traditional territory the university stands, and the Songhees, Esquimalt, and Wasanich peoples whose historical relationships with the land continue to this day. I do want to start by introducing my colleagues. This is a huge, extensive group effort, as well as the collaborators working on this project, and then I'll take us through some basic herring biology, and then I'll focus more on the Strait of Georgia herring population specifically, citing some of the work that PSF and UVic has conducted, which really highlighted the importance of Strait of Georgia herring to Strait of Georgia salmon. So as I said, it's a highly collaborative project. We're working closely with researchers at 3 labs at the University of Victoria, several First Nations communities around the Salish Sea, researchers from Environment and Climate Change Canada and DFO, as well as private environmental consultants and NGOs. At the Pacific Salmon Foundation is Isabel Persall, Director of the Marine Science Program, Will Dogood and Katie Innes, both biologists on this project, and myself as the project manager. At the WANIS lab at UVic, we have our field coordinator Avril Hahn and research technician Maddie Thompson, soon to be WANIS lab postdoc Rhonda Reedy, and then 3 master's students, Neil O'Brien, who's at Royal Roads, and then Loic Dallaire in the Costa lab, and Jake Dingwall in the Bates lab, both master's students at UVic. Okay, so now let's talk about herring. I am preaching to the choir when I say this, but we should pay attention to forage fish because of their position in the food web. Forage fish eat plankton at the bottom of the food chain and transfer that energy up to all the marine mammals and predatory fish like salmon. So changes in forage fish abundance, changes in where and when they occur, can tell us a lot about how those components of the ecosystem are connected, how that ecosystem functions. Many of us are familiar with Pacific herring. They're found from Baja California to the Bering Sea and over to Japan. They're a coastal species associated with the continental shelf, inlets and estuaries, and in our region, most are sexually mature around age 3. But unlike salmon, they can spawn year after year. They are repeat spawners, some living up to 10 years or more. In fall, adult herring leave summer feeding grounds on the continental shelf, and they move inshore to overwintering areas where they mature and prepare for spawning in spring. And the timing of those spawns is genetically controlled. So in the Strait of Georgia, most herring spawn around end of March, around early February into March. But some genetically distinct stocks in and around Puget Sound spawn anywhere between January and May, January to May. Spawning occurs in intertidal and subtidal habitats. Females deposit small sticky eggs that adhere to vegetation and substrate, and then males release milt, which is what causes that beautiful turquoise color that is literally out of this world. You can see these plumes from space. Or from a drone. This image was taken by Loic Dallaire off of Qualicum Beach, and you can see that these really are just big feasting events. They attract predators like birds, seals, sea lions, as well as those predators, such as transient orcas. So it's really a massive pulse of energy to the marine food web. And then depending on temperature, eggs hatch after about 2 weeks, at which point the larvae are extremely vulnerable to environmental conditions. A lot of mortality happens at this stage, but those that survive school in nearshore habitats like kelp beds, seagrasses, and even around your local marina dock. And so this is the stage when they are really important to predators like salmon. And then at some point over the next 2 years, they join the adult population, although when and where they join adults is unknown, but we know that adult schools tend to contain a mix of different age classes. Historically, BC herring have supported a number of important fisheries. First Nations harvested adults directly and eggs through either row-on kelp or row-on boughs. However, the loss of spawns in some territories, particularly in the southern Strait of Georgia, have become a concern by First Nations that have lost cultural ties because they cannot access herring resources. Unlike salmon fisheries, which decline on a sliding scale, herring fisheries are either open or closed. And that's based on whether there's enough spawning activity in that area to support that catch. So if there is no spawning, there is no catch. Back to the 1960s, there was an intensive reduction fishery on our coast, as Graham pointed out, for herring oil and fish meal products, and that took up to 250,000 tons of herring per year, which ultimately led to a coast-wide collapse and a closure of the reduction fishery by 1967. As herring numbers started to recover, a new roe herring fishery started up, targeting mature females for their roe prior to spawning, which has become a major export to Japan. Smaller food and bait and special use fisheries were also developed to target adults for both human consumption, pet food products, and fish bait. And then since 2019, the Strait of Georgia population has been the last remaining stock still open to commercial fisheries. All other areas have been closed, although in the last 2 years, the Strait of Georgia harvest rate has been halved from what was historically 20% down to 10%. So in the Strait of Georgia, ecosystem herring are a key component of the salmon food web. We know salmon eat herring. There are several ways, but there are several ways they can interact. So juvenile herring can be important food for juvenile salmon that first enter the marine system as they boost growth and survival. Juvenile and adult herring are food for adult salmon throughout the year as they support salmon growth and reproduction. And then in some cases, adult herring can eat small salmon fry like chum and pink that enter the ocean at a very small size. Juvenile and adult herring also eat similar types of prey as juvenile salmon, so they may compete with each other for food. And then lastly, the presence of herring may buffer against the effect of pinniped or seal predation on juvenile salmon. So if there's lots of herring around, pinnipeds may shift their focus away from juvenile salmon and instead eat herring. So the point really here is that it's complicated, but there's several ways that herring and salmon can interact, many of which we don't fully understand. Yet food supply and predation were found to be key factors impacting salmon survival. So what you're seeing here is just an infographic synthesizing the findings of over 90 publications that came out of the Salish Sea Marine Survival Project. And that was a large-scale initiative that was partnered with Long Live the Kings to identify key factors influencing the declines in Chinook, coho, and steelhead survival in the Strait of Georgia. And so it's really those findings that are informing the herring work that is being conducted by the Marine Science Program at PSF. And so the Strait of Georgia herring population, as I mentioned earlier, is the largest and last remaining stock still open to commercial fisheries. Spawning is concentrated along East Coast Vancouver Island from Nanoose to Comox. It's believed that juveniles spend their first year, or at least their first year, rearing in the Strait of Georgia. After that, they migrate between spawning grounds in the Strait of Georgia and summer breeding grounds off the West Coast, Vancouver Island. And that's the way they're managed, under the assumption that all of them are migratory, except some adults actually remain in the Strait of Georgia through summer as non-migratory or resident herring, but we have no idea how many stay behind or why. Since the 1980s, both the geographic distribution of spawning events and the diversity of spawn times has declined. Over that same period, though, the abundance of the population has remained relatively stable. So it's not that a decline in spawn distribution and timing is because of a decline in abundance. It's more that everyone is now spawning in the same place at the same time. So this could have implications for herring availability to different predators like juvenile salmon. So if historically, there were spawns occurring in different places in different times of the Strait of Georgia. That might mean more feeding opportunities for juvenile salmon that first entered the ocean anytime between April and July. So a loss of spawn diversity could have reduced those feeding opportunities. And there is some evidence of this. So herring did make up a much lower proportion of herring currently make up a much lower proportion of diets than they did prior to the 1990s, that should say, when salmon survival was higher. And then more recent work by my colleague Will Duker during his PhD work in the Southern Gulf Islands has suggested the distribution and timing of spawning events may also dictate the distribution and timing of juvenile herring to different regions of the Strait of Georgia. It may influence when they arrive to those different regions and at what size. And so herring size is important to juvenile salmon because they're gape limited. So they can only eat things that are smaller than the size of their mouth. So the plot that you're looking at here is showing the size and capture date of juvenile salmon that are eating herring in the Southern Gulf Islands. And you can see that herring don't actually appear in juvenile salmon diets until late in the season, around mid-August. And it's only the larger juvenile salmon that are able to eat herring. So you can picture a herring that hatched in Qualicum in spring, which is where that yellow star is on the map. It may take all summer to arrive in places like Cowichan Bay, and in that time it's growing. So by the time it arrives, perhaps it's only the larger salmon that can eat them, which is bad news if you're a small salmon. But this is a really critical time for juvenile salmon that first enter the marine system, as their ability to grow right off the bat is increased if they have access to herring. I mentioned earlier that some adult herring are known to stay in the Strait of Georgia through summer as resident herring instead of migrating to the West Coast. This isn't something new. Resident herring were first documented in 1964, but we know very little about them because there's been no long-term monitoring of herring in summer. Anecdotally, First Nations and local fishers say they used to be quite abundant and available year-round until they disappeared in the 90s. And then recently, rec fishers have reported a reappearance of large herring aggregations at popular fishing spots in the Strait of Georgia. And many have said that salmon fishing in the Strait of Georgia is the best they've seen in over 30 years. And so I think the best thing we can say for sure is that resident herring tend to appear and then disappear from the Strait of Georgia. These recent reports are also consistent with data from the Adult Salmon Diet Program, and that is an ongoing program that started in the Wanis Lab at UVic and has since been funded by the Pacific Salmon Foundation. The program works with recreational fishers to sample salmon stomachs throughout BC to understand food web structure and to track changes in the ecosystem. So what you're looking at is the diet data of Chinook salmon from 2017 to 2023. Covering the 5 regions in that map in the upper corner, and they correspond to those 5 regions and panels in the plots. Each panel is showing summer diets on the top and winter diets on the bottom. Each bar indicates how full salmon are of each prey type in that given region, season, and year. Don't freak out. I know it's a lot to look at, but I just want you to focus on the green panel and the green circle, which is representing the Strait of Georgia. What sticks out is that Strait of Georgia Chinook are full of herring in summer. And then if we take those same herring, estimate their age and break it down by age class, Chinook are eating the larger adult resident herring that are age 2 and older that do not migrate to the West Coast, but stay behind in the Strait of Georgia for reasons unknown. These same resident herring have been the focus of my master's research over the past few years. So these are screenshots from our boat sonar during my fieldwork. The top 3 panels are from a site just south of Quadra Island, and the bottom 3 are just 20 kilometers south at a popular fishing area called Katie Coleman Reef. The dark line is showing The dark line at the bottom is showing the seafloor, and then everything above it in those blue, green, sometimes red bands are herring schools, sometimes reaching up to 90 feet thick in some areas. So there is always a myriad of predators associated with these aggregations: salmon, dogfish, humpback whales, and many seabirds. So these are very active feeding areas and likely an important source of food for these predators. But this wasn't necessarily the case 20 years ago. This same region was considered to be relatively unproductive when talking to local fishers that have spent a lifetime in this area. So the question is, what has changed? Why have resident herring, quote unquote, reappeared? So we should pay attention to resident herring because changes in their abundance may be reflecting shifts in the environment and at lower trophic levels that can trickle up through the food web. So there's lots of resident herring around. That could mean growth benefits for salmon and benefits for survival and reproduction. Fewer resident herring in another year could mean that it is a poorer year for salmon. So whatever is kind of driving these long-term residency patterns may also be driving food web dynamics. So then, given some of these observations through the Sailor Sea Marine Survival Project and through the UVic Adult Salmon Diet Program, we wanted to reach out to a broader herring community. In October 2021, PSF, along with UVic, Cowichan Tribes, and UBC, hosted a herring workshop to discuss herring research priorities. That brought together First Nations, community groups, and researchers to first identify the knowledge gaps and key questions related to herring, provide a forum for participants to voice concerns and summarize ongoing work. And then finally, that was intended to facilitate collaborations to pursue funding opportunities aimed to, address those key questions. And so that's what we did. We developed a proposal in collaboration with UVic and Cowichan Tribes and submitted that to the BC Salmon Restoration and Innovation Fund in November of 2022. That was successful. PSF was awarded $3.7 million to study Strait of Georgia herring. That project started last May, and then it extends until March 2026. And so we are just building that program up now to start fieldwork this coming May. And so I'm going to very briefly go through the 4 activities under this program. And what I just want to get across is that while the overarching goal is to address those scientific questions, an equally important goal is to pilot new methods to study herring-salmon interactions using small boats so that, citizen scientists like yourselves and particularly First Nations communities can be trained and equipped to continue this work if there's interest to do so. So these are the 4 activities. The first is led by my colleague, Will Dogood. That's investigating the interactions between age 0 herring and juvenile Chinook and coho salmon. I talked about this idea earlier. The objective is to understand if spawn distribution influences the dispersal of juvenile herring to different regions in the Strait of Georgia. Does that influence their availability to salmon in terms of herring abundance, the timing, and size? And then this is important because this could have implications for their early marine growth and subsequent survival. So there's a couple components here. Part of this activity will fund a master's student to analyze a historical data set from the Strait of Georgia age 0 herring survey and then relate it back to spawn distribution in those years. This activity also includes a field sampling program to non-lethally sample juvenile salmon and the juvenile herring that they're eating throughout summer in 5 different regions on the map there. And that's just to follow salmon through their early marine life using a combination of beach seining, purse seining, and micro-trolling. The second activity is to develop a biomass estimate and otolith-based detection of adult resident herring in the Strait of Georgia. And that's intended to measure changes in resident herring biomass and then to identify drivers of long-term residency patterns. The fieldwork component is going to use a scientific echo sounder, stereo camera deployments, and we will also be jigging for herring. And all of that data is integrated together to get an estimate of herring density in a surveyed area, which we can monitor annually. The herring samples are also kept for otoliths, which is an extension of my master's work. I've just blown up an otolith image in the corner there. The otoliths are used to differentiate residents from migrants, which currently we can't do just from looking at them. So for those who don't know, otoliths are these paired calcium carbonate structures that grow in a fish's head, and they help the fish hear and differentiate up from down, but they also grow like rings in a tree. So as fish move between different environments, they lay down different elements into those rings, which we can infer their movement patterns from. And that's important because if we can tell residents and migrants apart, we can start to ask questions about why they're different. What makes a resident, and why do some fish go while others stay? The third activity is looking at herring spawning habitat, and there are two projects that are being led by UVic master students, Loic Dallaire and Jake Dingwall. Loic is doing some really cool work on using satellite imagery to detect spawns. So currently DFO runs aerial spawn surveys to direct dive teams on the water to locations where the dive teams conduct egg surveys. But some spawns might be missed by those planes if spawns are short-lived, if they're deep, or if they occur outside of core fishery areas and periods. And so an alternative approach may be able to fill those sampling gaps. So Loic's developing a spectral spawn index, which essentially compiles a profile of different wavelengths of light that are reflected off spawns and are specific to spawns. So we can see in the visible light range, but there are several other wavelengths outside that range that are reflected off the milt. Then that index can be used to separate spawns from other similar looking white things like sand or sea foam that appear in those satellite images. The next component is work that Jake Dingwall is doing with MC Wright and Rain Coast Conservation to investigate spawn habitat quality. Herring seem to return to kind of the same key locations between Qualicum and Comox, but in some years they surprise us and they spawn somewhere totally different, and we don't know why that is. So Jake's work is going to survey spawn habitats that are persistently used versus those that are more intermittently used to compare differences in their temperature profiles, vegetation type and cover, contaminant loads in both the sediments and the water, as well as the proximity to nearshore developments like pulp mills or impacts from vessel traffic and noise. And then the last activity is led by Isabel Persall, and this is a First Nations community-led outreach initiative. What the herring workshop really highlighted was a lack of herring resources available to First Nations, and because of that, a lack of knowledge transfer from elders to First Nations youth on how herring were traditionally used and prepared. So the goal here is to provide funding to First Nations partners to lead and organize community workshops and events as a place to share that knowledge through videography, educational materials, or art, which is produced by and developed by First Nations videographers and artists. So it's really meant to provide funding and products for the community to celebrate herring, rebuild those connections to herring, and hopefully inspire the next generation of marine stewards. So I'll finish by saying that herring are the backbone of the Strait of Georgia ecosystem. All forage fish are, but we're excited to learn about how herring may influence food web dynamics in the Strait of Georgia and how they support salmon survival. And we hope this project can be a stepping stone to continue this work. I think a success in our minds would be if we develop these methods to a point where others could carry the torch, so to speak. So I'll leave it there. And I'm happy to take any questions. My contact information is there if you have any further questions or wanted to reach out to any of the team members. So thank you.
Right on. Thank you so much, Jessica. What a fantastic presentation. Amazing to see all the work that you're doing in your own research and collaborating with others and the Pacific Salmon Foundation, how many facets there are to this project. It's really incredible, all the different angles that it's covering and progressing the knowledge of herring further. Without further ado, there's a couple of questions in the chat. One from Ramona, who is asking about your bar graph slide, referring to the diets of salmon. The year is shown for each data set, but what time of year are your salmon being sampled? Is it across all seasons? With the kind of note that there would be different seasonal abundances at different prey species.
Yeah, so the years were 2017 to 2023, and this is aggregating all of that data. It's a measure, it's a partial fullness index. I didn't prepare these data, so I'm trying my best, but it basically samples come in that are captured from different areas in different months. And the goal is to try to source stomachs from recreational anglers to get a good spatial and temporal picture of the prey forage fish and what they're eating. So we try to avoid going to derbies, which may kind of up the sampling number in a given region and season and year. Is that answering your question?
I think that answers it. And
it's
Ramona,
tough though.
Yeah.
It's tough though, because you're relying on, fishermen going to the hotspots, and then, you get fishermen that are really enthusiastic. So it's kind of hard to manage things in that way. It just kind of gives a broad perspective of what's going on when we didn't know anything at all.
Yeah, I think that's a great answer, and it covers it. And for it, where can people find out more about the salmon diet study, Jessica?
Yes, yeah. I wanted to mention that. Contact myself and I can link you up with the adult salmon diet program manager who works at UVic.
Oh,
I don't have his email right off the bat, but here I will contact me.
It looks like Will's chimed in the chat there. We'll do good with some more supplementary to that question.
Okay, perfect. Thanks, Will.
I see a hand.
He's spearheading the ASDP, so he's the best one to ask.
Yeah, and then we have Teresa. Is your hand up? Is that a fresh hand? If you like,
yes, it is.
Cool. Yeah, go ahead.
I have one quick thing. I wondered if that iNaturalist herring spawning spotter program would be helpful to Jess's research for finding those small spawns. And then the other quick thing is, do you have any sense that the resident herring that hang around are somehow a leader in for the migratory stocks that are coming in to spawn? Is there any kind of thought about that?
A leader, sorry, could you repeat that second question a bit? A leader, so they're leading the migratory stocks
into the best herring, the best spawning areas. Like, what's the kind of relationship between those two populations?
Yeah. Okay, so the first question, I'm bad at two-parters, but the first question, yeah, I think the iNaturalist kind of boots on the ground observations are really important when weather is bad. So, similarly for the aerial surveys and for the satellite imagery, if it's cloudy, it's kind of no good. So that's where the citizen scientists come in, is that they can fill in those weather sampling gaps. So I think that would be really useful in that way. Also, so the second one is, how are migratory and resident herring related? I mean, they are, they belong to the same population, but whether it's just a migratory fish or a resident fish, we don't know. Fish could start off as being migratory and then become resident, or vice versa. So there isn't really a lot known about the degree of different sort of migration strategies that exist in the population. I don't know the answer. I'm trying to figure that out.
Yeah, thank you. And I think you're right when you say, you say the importance of herring. Herring, it's crucial to the health of the whole ecosystem in the Strait of Georgia. I think you're spot on.
We've got,
good. I agree. Thank you.
Some other questions happening, but maybe we can carry forward. We don't want to sell Simone short here. But thank you so much, Jessica. Fascinating work, and we're looking forward to updates as they come along. Clearly, there's this so much information to learn about these species and their ecology. So thank you so much, Jessica, for sharing that. That was awesome.
Thank you.
Our next speaker, Dr. Simone de Roches, began a research career at the University of British Columbia, studying how intrapacific variation within three-spine stickleback fish affect their lake ecosystems. This is a great origin story and background for, I hope she comes out with a book that goes through all of her research. But during her PhD at the University of Idaho, she studied the evolutionary ecology of and natural selection on lizards and white sands. After moving to California, Simone did her postdoc at the University of California, Santa Cruz, and explored stickleback's rapid evolutionary responses to climate change in California estuaries across a latitudinal gradient. She made it back to her favorite bioregion for 2 more postdocs at the University of Washington. The first is part of an international collaborative network studying urban evolutionary ecology, and the second with the wetland ecosystem team in the School of Aquatic and Fishery Sciences, developing and analyzing from the Shoreline Monitoring Database, which I hope some of us are familiar with, a community science-based repository for standardized protocols and data on effects of shoreline restoration in Puget Sound. Simone joined the WDFW Habitat Program as a research scientist nearly a year ago and is examining relationships between fish, particularly salmonids, and their habitats. She is broadly interested in exploring how habitat modification and restoration affect the intraspecific variation and evolutionary trajectories in salmonids and other fishes. Simone lives in Edmonds with her husband, nearly 2-year-old, and senior menagerie of a quichond, oh my goodness, 2 rabbits and a crested gecko. She is passionate about art and science and loves watercolor, knitting, natural dyeing, and pigment making. And I'll just add, if you Google her and go to her website, she has a wonderful webpage about herself with a lot of these kinds of things featured, including her research and artwork. Please welcome Simone. Take it away, Simone. Welcome.
Thank you so much. Thank you so much for inviting me to talk with you this evening. I am really excited to be talking to folks in BC, as that is my home, and I'm excited to share some of the work I've been doing in Washington. So this talk is a little bit more general or broad than forage fish, as you might have guessed from the title, which is ecological variability as an indicator of shoreline restoration success. And but you will probably be very aware of and realize and notice that all of the habitats I'm talking about are really essential for forage fish. And I'll point that out in a couple different places within the talk, but you will recognize all these things that we measure as being really important for our forage fish. So with that, I will start, and you can all see my screen okay, I hope. Awesome. Okay, so I want to start with this very basic premise that we as humans have a way of really simplifying our landscapes. And this is especially true in our urban and urban adjacent areas. We tend to take really complex natural ecosystems and we homogenize them, we make them simpler. And this usually manifests as introducing non-diverse exotic species that make up monocultures like lawns, by creating impervious surface, by paving roads. And this not only reduces the complexity in given habitats, but it also makes different habitats in different places more similar to one another and really less variable through time. So in other words, they're less spatially and temporally variable.
Big fish bags that didn't.
And we do this in nearshore ecosystems too. We take complex heterogeneous shorelines that are really craggy and different, and we construct hard armor like bulkheads and seawalls. So we effectively take these transitional ecotonal habitats that many of you work in, and we make them flat and simple. We gave them this hard boundary. So here down in Puget Sound, nearly a third of our shorelines are armored, and this armoring is shown in red. This is a map created by our Department of Ecology. This makes up around 700 miles or 1,100 kilometers of these artificially simplified habitats. So it might be obvious why we pave roads and plant lawns, but why do we armor shorelines? And I think somebody alluded to this in an earlier talk, and I'll get into this in a little bit more detail. So let's zoom into a cross section here, and we can examine why we armor shorelines. So typically, at least historically, we've put up armor to prevent erosion. It stabilizes sediments at the shore that typically loosen due to wave action, storm surges, and rain. And so this means we can build and construct our structures, our houses, our buildings right up to the water's edge. But as many of you will probably be aware of, armor really disturbs our shoreline ecosystems at both large and small scales. And as I alluded to, it removes this physical and biological complexity and simplifies the shoreline ecosystem. So living in and around BC's coast, you're probably aware of what shoreline armor looks like, but in case you are not, I'll give you a few examples. These are from around Puget Sound. We have examples of shoreline armor in the form of riprap, which is basically essentially rock rubble on the left-hand side, and shoreline armor that's more like seawalls and bulkheads on the right-hand side. And these are usually made of concrete or wood, and they're varying sizes and lengths. So I'm going to walk through some visual demonstrations of what happens when shorelines are armored versus when that armor is removed and the area is restored. So first, armor, especially when it's built low on the shore, prevents beach wrack and driftwood logs from being left behind with the receding tide. It's usually built over natural dunes and sediments, and these are necessary for the establishment and the growth of riparian vegetation. So therefore, we have less vegetation in these armored shorelines. And because their logs, the wrack and the riparian vegetation provide habitat and nutrients for other organisms like insects, we often see fewer of these in armored shorelines as well. And then fourth, sediments are often more uniform in size rather than being a more dynamic mix of both gravel and sand. And because y'all are very interested in forage fish, I've highlighted some really important things for forage fish here. So one of the major ones is that we are basically taking away that shallow water habitat that is essential as a protective refuge area for juvenile salmonids to rear and forage, and for forage fish to also do those things and to lay eggs. We also, in many cases, have completely removed this egg-laying habitat for forage fish. And when it does remain, it's often more exposed to sun because we don't have that vegetation. And it often has this uniform layer of sediments that is less ideal for egg laying. So essentially, armor removes this shallow water intertidal ecosystem and abruptly transitions these areas, these shorelines, from terrestrial to marine habitats. Together, all these changes mean that we have decreased connectivity between our terrestrial and aquatic ecosystems. So in natural shorelines without armor, wrack and logs can accumulate on this naturally sloping beach. Vegetation can establish on the backshore, and we can have juvenile and small fish like forage fish that can forage and escape bigger fish in the shallower waters. We can also have wave action that breaks down sediments, making them more heterogeneous and different and diverse. And this all creates habitat for organisms like invertebrates and vertebrates. So one of the primary aims of restoration and that armor removal then is to get back to this naturally functioning ecosystem. So how have we monitored the effect of armor and its removal across Puget Sound? So we've partnered with a lot of local organizations, including the Vashon Nature Center, the Northwest Straits Foundation, and I had this talk before I joined WDFW, but also with WDFW and a lot of community scientists volunteers to monitor over 100 sites in Puget Sound. And so today we'll focus on a few studies that use data from 20 to 30 of those sites. So most of our sites have 3 different beach types or treatments from which we can compare different ecological responses. These are pictures of Howarth Park. On the left and bottom is our natural reference site, which has never been armored. In the center is our currently armored beach, which has not been restored. And in the right and top, we've got our restored beach where that armor has been removed. And also in this particular case, native plant species have been planted as well. So on our website, we have many different standardized protocols for measuring lots of different biological and physical responses to armor and restoration across these 3 different beach types through the years. This is a screen capture from our website, which I will provide a link to at the end of the talk. And notice that forage fish eggs is one of these. This protocol is from the Department of Fish and Wildlife again. However, I haven't personally analyzed those data yet, unfortunately. So the 3 studies that I'll present some results from collectively focus on beach rack, beach logs, supertidal insects and invertebrates, and sediment size. Okay, so how do we and community volunteers measure these variables? So the first step is we lay down a 50-meter transect parallel to the shoreline, and we measure our response variables at intervals along this transect. So broadly, we measure beach rack using a quadrat, as shown here. Here, from this, we can approximate the percent total cover of rack and the percent of different types of rack, so terrestrial, algae, and seagrasses, and we can also measure that rack depth. We also measure logs by laying a perpendicular transect to the shoreline, and we count the number of logs crossing that transect. And we also count the width of the log line, and that's the distance between the first and last log perpendicular to the shoreline. We also categorize the size of logs, so whether they're large or small. And then finally, we measure insects and other invertebrates using this fancy contraption that is a fallout trap. Just kidding, it's basically just a tub of water with a little bit of soap, and many of you have probably used these before. So we put this out in our habitats one day, and we go and collect them the next day. We filter those insects and invertebrates out, we preserve them, and we bring them back to the lab to count and identify. So from this, we can estimate the abundance or the density of insects and how many different types or their richness, the number of families that are represented. And then finally, we also look at sediment size, and this we broadly characterize as either gravel or sand, so smaller sediments or larger sediments. And again, we estimate the proportion of each of these using a quadrat. But here we do it at both the surface and subsurface levels. So overall, with this first study, we wanted to know how restoration affects these different ecological responses. We generally expected that the abundance of rack, of logs and invertebrates to decrease with armor, and hopefully again increase when that armor was removed and the site was restored. We also expected the diversity, like the number of different rack types, the richness or number of families of invertebrates, to also decrease with armor and then again increase with restoration. So without further ado, here are some of our results. These are basically done with before-after control impact design, which is a mouthful. People call it a BACI design. Basically, all this means is that we're looking at different abundances responses on the y-axis, our beach rack, our beach logs, and our supertidal invertebrates. And then on the x-axis, we have our before and after. And so this refers to before and after restoration. It's important to note here that this only applies to that blue line endpoints, which is our restoration treatment. Nothing actually happens in the green and the gray. The green refers to our natural reference sites, and the gray refers to our armored control. So let's zoom in a little bit here. What we expect to see is that if restoration is effective, our response variables will be more similar to our the restoration site will be more similar to our armored control in the before situation, and hopefully more similar to our natural site at the after time period. And indeed, this is fortunately what we see with many variables, as we can see here, pretty noticeably with beach logs. We also, as I said, looked at the diversity of rack types and the proportion of different size logs, so number of large and small logs, and in the invertebrate richness of families and orders. And again, in many cases, we see an upward trend in those restoration sites before and after. And somewhat interestingly, for a lot of these, we also see a downward trend for that armored site, those armored sites, which kind of suggests that maybe we're picking up on a pattern of decreases through time, like a bigger pattern of decreases through time. So these backy designs, the before after control impact, typically just look at the averages or the mean changes before and after restoration. And less commonly do they look at the actual variability around those means as a measure of restoration success. And this variability can really be illustrated using these box plots here for each treatment. And so again, zooming in here, what I want you to pay attention to is that when these box plots are super big or they're tall, that basically represents more spread in the data, which you can see with the data points here too. So the more spread in the data, the more variability we have at that particular treatment. And so in most cases, in our armored controls, we actually have much less variation than in our natural reference sites. For our restoration treatments, in this case, for beach rat cover, we see comparable variation to our natural reference. But if you look at through all the different variables we measure here, usually it's somewhere in between that armored and between the armored levels and the natural reference levels. So what was the source of this variability? Why did we see so much spread in the data? So this again is a little bit of an overwhelming plot, but I'll walk through it. This basically shows I've chosen 3 different variables here: beach rat cover, the number of beach logs, and the density of those invertebrates. And this is plotted through time from 2015 to 2020. And so, and then again, those colors represent our natural reference, our restoration treatment, and our armored control. But here they're plotted separately. So what I want to emphasize here is that the darker points represent our averages, whereas the fainter ones represent our individual sites. So you can get an idea of where that variability is coming from. So zooming in for beach rat, if you want to get an idea of variation over time, that's the amount of fluctuation or change as you move across the x-axis, whereas variation over sites is if we look at those error bars or the spread in the data at any given year. And if figures and graphs aren't your jam, here are some photographs of what that looks like. So here are a couple of one site rather in 2 different years. So this is Bowman Bay up by Deception Pass, if you've ever been there. And in one year in 2020, we have not so much beach rat. And in the other year in 2021, we have a lot of beach rat. Whereas here we have variation over space. This is again Bowman Bay in 2020, but compared to another site in the very same year. This is Edgewater Beach. So here we have not as many logs. And I know the angle of the photo is a little bit different, but in Edgewater Beach, we have a whole bunch of logs. So it may be hard to kind of decipher that in these graphs, but in general, the take home is that our analysis showed that there was more variation across space and across different sites than there was variation across time from year to year, although we had some responses that varied over both space and time. This variability is much less pronounced in our armored beaches over both space and time. It's almost as if that armor has really stifled or compressed the variation across the years and across sites. So it's kind of removing the highest levels, the areas with the most beach rack or the most logs or et cetera. Whereas that variability is much more pronounced in both natural and restored beaches. Okay, so why do we have this variability, particularly from site to site? So this kind of inspired a couple more studies that we did that I'll go over more quickly. The first was, first explanation is if you look at the different physical characteristics of the beaches. So where they are situated, what the geography looks like around them, what's the hydrography, sorry, where that beach is situated. So in this particular study, we looked at the different shore types. We looked at feeder bluffs, accretion shore forms, and pocket beaches. I'm not going to go into the details of those or the results of that study here, but I can show you a link to that. Suffice it to say that, you know, some of the variation comes from the different forms of these beaches. We also looked at fetch, which is the distance waves are generated by wind. And of course, that varies quite a bit in our area down here in Puget Sound. This is a really important governing force in the supply and movement of sediments, rack, and logs. And so what we might imagine is the more fetch or the bigger the fetch, the more stuff that's accumulated on the beach. So beyond the geophysical features, if we think about our restoration sites in particular, the actual characteristics of restoration, how it happens, can also lead to differences among sites and across years for just those restoration sites. So sometimes when we do armor removal here, there are supplemental restoration actions, and these include three main ones, which are sediment nourishment, or we're adding sediment into the area after the armor is removed, vegetation or native vegetation planting, and log placement, which often even involves the anchoring of logs to a beach. So here's a couple of photos of some restoration sites before and after armor is removed. On the top, we have Big Beach on Vashon Island, where no supplemental restoration actions occurred. And on the bottom, we have Cornet Bay and Deception Pass again, where we have armor removed, but also we had native vegetation planting, log placement, and sediment nourishment. So as with lots of restoration, it can be notoriously difficult to monitor the effects of individual restoration actions. And this is because they're rarely carried out in a controlled manner, independently or randomly across sites. It's not really an experimental design, or there's not really often an experimental design behind it. It's more opportunistic or depends on funding and logistical constraints. So for example, in our sites, of the three actions I described, only vegetation planting was ever performed independently or alone and without the other two, nourishment and log placement. So for these variables, what we found was that log placement in general, when we looked at all of them together, was the most important driver of differences in our ecological response variables. So sediments, log count, this is logs that actually accumulate, and beach rack. So in these plots here, just very generally, we have our gray bars, or our gray box plots showing our before armor removal. The blue shows armor removal without logs placed, and then the brown there represents armor removal with logs placed. So it's important to note that though log placement was the most important, and I can answer any questions about the stats there, but this does not mean that other additional actions were not also important or effective. It's just we could not, because of the lack of experimental design, really decipher or isolate the effects of those actions in and of themselves. So like I said, models with log placement was consistently the most important. It influenced and increased the proportion of surface and subsurface gravel, the number of additional logs, and the amount of beach rack and cover. So one thing that was interesting was that beach rack depth continued to increase in the years following restoration when you had logs placed, but not when you did not have logs placed. So it was almost as if these logs were causing the continued accumulation of beach rack through time. So these last two studies showed that both geophysical and restoration characteristics can really influence this variability across sites. And collectively, this suggests that there's a combination of social, geological, and ecological factors that could maybe interact to basically cause this variation among sites and across years in both our restored and our natural shorelines. And my plug is basically that it's important to measure this variability. And somebody mentioned that in talking about the variability in herring spawning as well. And I think that's totally true, because this is what can have a multifaceted effects on multiple levels of the ecosystem and of our society. So for example, variability at one ecological level can affect variability at other levels. And these can promote biodiversity and ecosystem stability and productivity, as I'm sure you are very aware of in forage fish examples and differences in breeding times and places. Further, it can be really important for humans too, because it increases the abundance and the reliability of the species we harvest, like salmon and shellfish, and in forage fish as well. But the last thing I want to emphasize is that our view on this variability is quite complicated. For some people, this variability in restored and natural shorelines is captivating and interesting and engaging. So every beach changes a little bit every time you visit it, or you go to different beaches for different experiences, for bird watching, etc. For others, however, and we've heard this before, restored and natural shorelines seem messy, they're unpredictable, they seem unmaintained. And this is particularly the case in the beginning. When we remove armor, things look super chaotic and kind of like a catastrophe, right? This is Docton Park, which is on Vashon Island again, before and after armor, like immediately after armor removal. So you can see that all these red alder trees have just like fallen to the ground without that retaining wall. But these alder trees are going to become logs, and that's important to habitat. So I want to close with the idea that restoration of shorelines and of other habitats as well might increasingly encourage the recovery of variability and measure this variability, and also encourage the appreciation of this variability from humans. And so projects like some of yours that involve community scientists are really great for that, because they're a great way to share that messy and chaotic shores are also a really good sign of a functioning ecosystem. So just to finish off, here's a QR code, which I thought was a fancy way of showing you where our website is, which is also pretty simple. So maybe it's kind of overkill, but the data I was working on here were all uploaded to our website, the shoremonitoring.org. And these are uploaded by many different groups that I mentioned previously that coordinate our community scientist volunteers throughout Puget Sound. And this website includes field protocols, data upload instructions, and downloadable data and some preliminary visualizations as well. So with that, I will end there, and thank you so much to all our contributors to our project and our funding from EPA especially, and yeah, the folks that collect the data and coordinate the fieldwork, because that's really what drives projects like these. So thank you so much.
Yay! Thank you. Oh, that was a great presentation. Beautiful slides. I imagine those are all your artwork, which is
They are indeed.
fantastic. There's a couple of questions in the chat, but I want to ask one first, and I'm going to. I'm curious about. Oh snap! Did I just forget my question? Oh goodness, I hope not. Okay, we'll move on to someone else's question. We'll do Jacqueline Mars. Jacqueline asks, how do you and/or your colleagues work with communities and private landowners to remove hard armoring and restore beaches?
This is such a great question, because a lot of our examples here are not of public lands. They are private property. And unlike in Vancouver, I think I'm not sure if it's just Vancouver, but like in Washington at least, we have a lot of shoreline that is privately owned by homeowners. So I don't think that's as much the case at least not in Vancouver where I grew up. It's, mostly public. And so that was really, I will say also that's a very different thing for me. But to answer your question, we have a lot of programs. One of them is called Shore Friendly. And I don't know if that logo is here somewhere. Shore Friendly, Friends of the San Juans. These programs, these groups can provide grants to help folks actually remove their armor, or in some cases, I have a picture of one of these where folks actually moved their house back. So obviously this is a very privileged thing to be able to do. And not everybody, but of course, if you have a house on the shoreline, anyway, it doesn't matter. But my point is that there are actions that folks can take, and there are grants that people can apply for and funding that people can apply for to encourage them to remove this armor. But it's an upward battle, because not only do we have armor that's still being built, but we have armor that's being maintained. And we have at DFW a lot of different permits that are required for that. But when it comes down to it, you're probably going to do what your neighbor does. And so you're not going to have one little stretch of armor that's removed when your neighbors have armor on either side. So it's a really challenging question to answer and problem to have. And there's just, we do what we can, I guess.
That's a great answer. I did remember my question. It was
great.
about the modules, and you focused in on those three. And I'm wondering if that's because those were the most consistently monitored by all groups, or why you selected those. And then also, if you could do the whole study again, would you suggest those three parameters again?
Yeah, so that's a great question. And your presumed answer is correct. So yeah, they are the most consistently monitored responses. They're pretty easy to monitor. The lab I worked in at UW, the only one that seems a little bit more complicated might be the invertebrates. And actually the collection is really easy, but of course you have to have taxonomic specialists who can identify those invertebrates and count them, and you have to have the lab equipment. And so the lab that I worked at UW did all that stuff. So they were basically a repository for all those samples, and so they were able to do that. But stuff like counting logs and visually inspecting rack and gravel, those are all things that folks can do with really limited supplies and equipment that are really cheap just in the field. But we have other protocols that we're trying to do that involves a little bit more outreach and education, like doing things like bird counts and that sort of thing as well. I'm less involved with this project now that I've switched employers, but it is, we're constantly looking for projects that folks can do with limited resources, I guess. So that is why. And then the other part of your question was, what other things that would I choose to monitor? And there are lots of them there. If I were to choose to go back right from the beginning, as you heard in the beginning intro thing, I am a herpetologist by training and deep down in my heart. And so some of these sites, we actually have fence lizards, and I would love to look at fence lizard responses to armor and armor removal across where they exist in Puget Sound, because I think they're telling us something about the fragmentation of our shoreline habitats. So that's my nerdy answer. I'm sorry, it doesn't have to do with fish, but I am a herpetologist through and through. So.
Oh, wow. Yeah, that sounds like a herpetologist answer.
Yep.
Well, thank you so much, Dr. DeRoche. That was a wonderful presentation and super helpful for a lot of our groups. I'm going to pass it back to Caitlin. I know people are checking out, but she's going to bring us home. Thank you again to everyone.
Awesome. Thanks so much, Kyle. I just want to give a really big thank you to all of our presenters. It was so great to learn from all of you tonight. And I just want to say personally, I have a lot of hats that I wear with my job, and co-chairing this network is one of my very favorites. So a big thanks to all of you for making this such a pleasure and for facilitating so much learning and so much research. It's really wonderful. So a big thank you to our financial and in-kind contributors, obviously all of our volunteers and community of supporters, all of our members. We're here because of you and we're here for you. So thanks everyone so much. Jack is going to pop a feedback survey into the chat box. So if you have some feedback for us about the symposium, maybe stuff you'd like to see next year, stuff we did well, stuff we can improve on, please let us know. The feedback will be integrated into next year's symposium and to what we do going forward. So please give us a shout. And thank you all so very much. This has been a pleasure, and I love learning with you and alongside of you and from you. So thank you so much. And on behalf of the Coastal Forage Fish Network, please join us again next year and follow along with our work. Thank you so much and have a great night.